Lab 8, Carbohydrate Catabolism, Cellular Respiration by Emma and Bobby. So what is cellular respiration? Cellular respiration is the process by which living cells obtain energy from organic molecules such as sugars, fats, and proteins, and release waste products. The goal of cellular respiration is to produce ATP and NADH while also limiting the loss of energy as heat. And when oxygen is involved, it is called aerobic respiration, and the energy intermediates used are ATP, NADH, and FADH2. And the three main stages of cellular respiration are glycolysis and the breakdown of pyruvate to an acetyl group, the citric acid cycle, and lastly, oxidative phosphorylation. So stage 1 is glycolysis, and glycolysis occurs in the cytosol, and it is when a 6-carbon glucose is oxidized into two 3-carbon pyruvate molecules. And glycolysis occurs in 3 phases. The first phase is the energy investment phase, when 2 ATP are hydrolyzed to create fructose 6-bisphosphate. The second phase is the cleavage phase, when a 6-carbon glucose is oxidized into two 3-carbon molecules of glyceraldehyde 3-phosphate. And finally is the energy liberation phase, when 2 glyceraldehyde 3-phosphates are oxidized into 2 pyruvates, producing 2 NADH and 2 ATP as a result. So the net yield of glycolysis is 2 ATP and 2 NADH. So here's a brief overview of glycolysis. We can see from this diagram that we can see the energy investment phase occurring first. We start off with a 6-carbon glucose, and that turns into a fructose 1,6-bisphosphate through the addition of ATP and substrate level phosphorylation, which is when an enzyme directly transfers a phosphate from an organic molecule to ADP. So we can see that in this case, the phosphates are removed from 1,3-bisphosphoglycerate. So that's right over here, occurring in step 6, and phosphophenol pyruvate, which occurs in step 9 over here, and that is when the phosphates are transferred to ADP. Continuing on with glycolysis is pyruvate oxidation, and pyruvate oxidation occurs in the mitochondrial matrix, and it is when 2 3-carbon pyruvates that are produced at the end of glycolysis are oxidized by the enzyme pyruvate dehydrogenase. And a CO2 molecule is removed from each pyruvate throughout this process, and the remaining 2-carbon acetyl group is attached to a coenzyme A, or CoA, to make acetyl-CoA, which we can observe right here in the diagram. And 1 NADH is produced for each pyruvate. So the net yield of pyruvate oxidation is 2 NADH, 2 acetyl-CoAs, and 2 CO2s. And quickly looking at the diagram over here, we can see that pyruvate is made in the cytosol in glycolysis, we know, and it travels through a channel in the outer membrane and an H+ pyruvate symporter in order to reach the mitochondrial matrix of the mitochondrion. The second stage of cellular respiration is the citric acid cycle, and this occurs in the mitochondrial matrix as well, and it is when the 2-carbon acetyl groups formed from the previous pyruvate oxidation is removed from acetyl-CoA and attached to a 4-carbon oxaloacetate in order to form a 6-carbon citrate, or also called citric acid. And these organic molecules are oxidized in order to liberate 2 CO2 molecules per pyruvate. So as they follow around these 8 steps, as they go through this cycle, they become rearranged and oxidized in order to form back into the oxaloacetate as they did in the beginning. And so the net yield of the citric acid cycle is 4 CO2s, 6 NADHs, 2 FADH2s, and 2 ATPs, which are made through substrate level phosphorylation from GTP, which we cannot see on this diagram, but it's on the one in the textbook in greater detail. Oxidative phosphorylation is the third stage of cellular respiration, and it occurs in the inner mitochondrial membrane, and it involves the electron transport chain and ATP synthase, which we will be looking at in greater detail in the next few slides. And oxygen is the final electron acceptor in oxidative phosphorylation, and the net yield is 30 to 34 ATP are produced per glucose molecule that is broken down into CO2 and H2O. So let's look at the electron transport chain in greater detail. So the electron transport chain oxidizes or removes high-energy electrons from NADH and FADH2 and pumps their H+ ions into the intermembrane space, which is over here. So we can see that as NADH goes to the first protein complex, which is NADH dehydrogenase, it is oxidized to NAD+ and H+, so the H+ protons move through the protein complexes. We can see it all here. They move through, while the electrons, which is shown through the red line, move down through the chain, so the electrons pass through the chain of protein complexes, which I just stated, and so the movement of these electrons generates the H+ gradient and proton motive force. So when the electrons pass through the chain, this creates the electrochemical gradient of the H+ ions in the intermembrane space. So this results in an excess of positive charges outside the matrix of the mitochondria. So now what does that electrochemical gradient do? So this is where we turn to ATP synthase, which is the last step in cellular respiration. And so ATP synthase, which is located at the bottom, or is the last step of the electron transport chain, ATP synthase uses energy created by the H+ electrochemical gradient in order to pump ions across the membrane and phosphorylate ADP to synthesize ATP. And this process is called chemiosmosis. So H+ ions will move down their concentration gradient through ATP synthase, which is this structure here, and into the matrix. And so when this happens, ADP attaches to ATP synthase along with an inorganic phosphate in order to produce ATP. And this is a rotary machine, and at the end of ATP synthase, it produces approximately 30 to 34 ATP as it spins around. So that is a very high production of ATP throughout the ATP synthase. So now let's focus on the role of enzymes. So obviously we know that they regulate the rate of chemical reactions within a cell. And specifically, they also regulate the rate of cellular respiration. So let's look at the first type of enzyme, which is a kinase. And this is an enzyme that transfers a phosphate group from an ATP molecule to a specific target molecule. And this is called phosphorylation, which we talked about earlier. And for example, we can see that ATP is phosphorylated to ADP and an inorganic phosphate. And an example of a kinase is a phosphofructokinase, which we will talk about next slide in greater detail. Another type of enzyme is dehydrogenase, and this is an enzyme that oxidizes substrates by removing hydrogen ions. So for example, NADH would be dehydrogenized to NAD+ and H+, and FADH2 is dehydrogenized to FAD and 2H+ ions. And we can see that this occurs in the beginning of the electron transport chain and other processes throughout cellular respiration. So let's elaborate on what phosphofructokinase does. So this enzyme is a kinase, and it is found in glycolysis. And what it does is it phosphorylates fructose 6-phosphate to fructose 1,6-bisphosphate. And it is a key regulatory enzyme in glycolysis because its step is irreversible. So if ATP levels within a cell are very high, the cell no longer needs to produce energy, so phosphofructokinase, or PFK, activity is inhibited by allosteric regulation by ATP. However, if ATP levels in a cell are low, phosphofructokinase is stimulated by ATP again in order to speed up the rate of cellular respiration. So overall, phosphofructokinase really regulates whether glycolysis will proceed and to produce ATP, or whether it will come to a halt and stop the production of ATP, depending on the levels of ATP within the cell.